and good evening and welcome everybody to Pint of Science Netherlands. So today we are number 6 in the event of 2021 for the Pint of Science. And obviously we're still online, but that's okay. But hopefully next year we'll be back in the pubs. But we've had some really interesting shows so far and we're really happy to bring you quite an interesting one this evening. So my name is Hannah, I am your host tonight. And on my other side, this side.
I am Nefeli. Nice to meet you. We are members of the Pint of Science, Hannah.
Yeah, and we work as volunteers here, and just a big interest of ours to bring what goes along in the scientific community to you guys. And tonight we have quite an interesting topic. So tonight we're talking about, can medical science do without animal experimentation? So obviously this is quite an interesting one. It is a bit dear to my heart as I work as an animal technician. So, but you know, it's something that I would quite happily see my job maybe go without. And definitely you also find some experience.
I actually did my PhD in neuroscience, and during my research I used laboratory mice and rats, and I really see the need to use animals in biomedical science, but I also felt the need to reduce using so many animals. So I'm very, very interested in this topic today.
Yeah, definitely. And I think it's how a lot of scientists in the field see. So tonight, we're talking about, actually the Dutch government in 2016 announced that they wanted to go mostly animal-free testing by 2025. And obviously we're in 2021 now, and that seems, not so far away. They have loosened that a little bit, but it's still definitely something that they're interested in. And if you look at the EU report that came out, interim report that came out last year, in 2017 alone, we, in the whole of the European Union, used roughly 9.3 million animals in scientific testing. So how do we get from there to, reducing the amount that we're using? So tonight we have 4 speakers that vary on their topics within the subject. So we will introduce them into the studio now. So Nefeli, would you like to introduce our first speaker?
Our first speaker is Laurens Witter. He's a brain researcher.
Hi, Laurens, welcome.
Hello, Laurens. He's a brain researcher at Wilhelmina Kinderziekenhuis at UMC Utrecht. He studies early brain damage in young children, for which he combines research on humans and laboratory mice. And he will talk to us about why animal use for medical research is crucial for understanding what happens in the brain.
Yeah, looking forward to it. Happy to be here. Thank you.
Great. And our second speaker that we have tonight, we have Alasdair Irvine. So he is a PhD working at the Pharmacology department at Utrecht University. Welcome, Ali.
Hi, guys.
And so
nice to be here.
In Ali's PhD, he is attempting to find in vitro models. So, not in an animal, models that can be used instead of animals. And he evaluates the ethical and economical arguments for applying them and to help innovators to come up with challenges using these new technologies in medical research. So yeah, we're really happy to have you tonight to learn about this part of the argument.
And our third speaker is Cyrille Krul. She's director of the Research Center for Healthy and Sustainable Living and a professor of innovative testing in life sciences and chemistry, but also she's a member of the initiative that is called Utrecht Transition to Animal-Free Innovations group. She will explain us how the group works. She will show us where the opportunities and obstacles lie and how these obstacles can be removed. So, hello, Cyrille.
Hi, Cyrille. Welcome.
Nice to see you all.
Yes, you too. And our fourth speaker tonight, we have Daniela Salvatori. So Daniela is a professor at the Comparative Anatomy and Physiology of the Veterinary Faculty in Utrecht. And she's also the head of the Utrecht Transition to Animal-Free Innovations Group. So tonight, Daniela is going to explain her part of the story about how the Utrecht Transition to Animal-Free Innovations Group, how they work, and discussing their core values, which are welfare, health, quality of research and education, but also sustainability for the planet and all of us that live on this planet. So welcome, guys. I think we have quite a broad panel this evening, so I think some really interesting discussions.
And we also want to encourage the audience to add comments in YouTube or Facebook with questions. During the talks, we can forward them to the speakers, and your question can be answered immediately.
And of course, we have to start with a cheers to Pint of Science. So cheers, everybody. Welcome.
Cheers.
Cheers.
And great. So we'll start with our first speaker. So Lawrence, you have the floor.
Thank you. All right. So yeah, thank you all and good evening. It's great to be here. I would like to talk about the animal use in scientific research today. And to start off, I'm not here to say that I'm for the use of animals in research. I think that nobody in his right mind would say that he's for the use of animals in research. But what I am for is that we have a very careful deliberation of when to use animals in research and what we are actually going to use them for. Now, I think a good way to start this is to look at what actually is an animal experiment and what does the law in Europe and what does the law in the Netherlands actually say about this. So there's, of course, a lot of rules and regulations surrounding animal use in science. And I'm just going to highlight the two, as I said. So the Dutch law defines an animal experiment as something that is experimental and has a scientific aim with an unknown outcome, or it has a means of obtaining an education. And in these procedures, an animal could suffer pain, fear, or permanent damage. Now, what does this mean in practice? Let's see a few examples. For example, you capture a bird and you put a ring on that bird to see where it's migrating in winter. Now, this actually is an animal experiment because it has a scientific question. You want to figure out where the bird is going to. And it is actually a procedure where the animal could suffer, in this case, fear, because you capture the animal that could induce fear in the animal. Now, whether it's invasive or not, actually, we're not doing anything to the animal. We're only putting a ring on its paw. That doesn't matter for the law. So the law only concerns whether it has a scientific aim with an unknown outcome and whether there's a potential for suffering of the animal. Another experiment would be, for example, when you kill an animal and you take some of the tissues of that animal to use in an in vitro test. Now, this is also a scientific experiment. There's an unknown outcome, and the animal that you use is actually permanently damaged. You kill it. So this is also an animal experiment, which might be a little bit counterintuitive, because when we talk about in vitro testing, often we associate this with animal-free techniques. But this can be a little bit confusing. Now, when are we actually allowed to do these experiments? And EU law is very clear about this. Basically, you are not allowed to do animal experiments unless, and there are three criteria that you have to fulfill to do an animal experiment. The first one is you have to have a scientific justification. So you need to explain why you want to do this and what the scientific gain is. And then you need to take this scientific gain, you need to deliberate whether that, you need to compare that to the suffering that the animal is undergoing. And then the scientific gain has to outweigh the suffering of the animals that are undergoing the animal experiments. And then the third criterion, and this is probably the most important one, you're not allowed to do an animal experiment if you can do or obtain the same results without using an animal. So if you can achieve the same goals without using an animal, you're not allowed to do the animal experiment. Now, what does this mean for my work? I'm a neurobiologist by training, and I've worked in places like the Netherlands Institute for Neuroscience, the VU University in Amsterdam, and now I'm working at the Wilhelminus Children's Hospital in Utrecht. But I've also worked abroad at Harvard Medical School and the University of Leipzig. Now, no matter where you go in Europe or even in the US, these three basic principles will adhere, and they will guide what we do with animals. Now, what kind of things do we use these animals for? And in the Netherlands, we can distinguish several categories of animal use in science. And these are numbers from 2019 for the Netherlands. You can see that well over a third of animals are used. The biggest piece of the pie here, fundamental research. Now, what is fundamental research? When we look at, for example, neuroscience, fundamental research would be figuring out how the brain is connected, which neurons connect to other neurons, how a signal is going from one neuron to another neuron, what proteins are involved in that. Those are fundamental biological questions. So that's fundamental science. And if you use an animal for that, it will end up in the statistics here. Now, the second biggest piece of the pie is the legally required testing. And this is actually animal use that is mandated by the government. If you want to introduce something to the market, let's say medicine or a product, you have to show it's safe. And sometimes the government wants you or mandates you to use an animal test to show its safety. And this is actually the second biggest piece of the pie here of animal use in science. And you can see it's around a little bit under a third of all animals used. And then we have translational science. Translational is really the one that is going to bridge the gap between the fundamental knowledge on the one hand side and the clinic on the other side. Now, in neuroscience, this might mean, for example, translational neuroscience is there's a certain damage to the brain. What would that mean for a patient? Or there's a certain genetic defect that we see in patients. How does that influence the functioning of the brain? And how could that explain what a patient is undergoing? So this really bridges the gap. Now we have two different categories here, education and animal protection. Animal protection, you already know. We talked about this already. For example, you ring a bird to see where it's migrating, and you do this to actually prevent poachers from killing an endangered species. You want to make sure that these animals are safe. Then you do an animal experiment for animal protection. And education is also quite easy to imagine. For example, a student in a veterinary school, they need to practice surgeries or procedures on animals sometimes. So that would be animal use for education. Now, my focus is on the cerebellum. And when you look at the brain, the human brain, you can see this big part. It looks a little bit like a walnut. That's your forebrain. It's very complex in humans. Then basically from this walnut, there's a stalk that is connecting the brain to the rest of the body. That's the brainstem. And connected to these two structures is something that looks a little bit like a meatball. And it's at the back of your head. It's basically when you put your hand on the back of your head like this, you're holding your cerebellum. And the cerebellum is really important for the regulation of movement. But we have also indications that it's important for the regulation of cognition or emotion. Now, when you look up the cerebellum, the cerebral circuitry on Wikipedia, or even in a textbook, you will see something like this. Information is coming in here on the left bottom side, and then it's processed by these red cells and the black cells here, and then it's integrated in these really big cells here, which are called Purkinje cells, the green ones. And then from there, it is sent to all kinds of other regions of the brain. And here in the background, you can actually see these Purkinje cells. This here, this darkly colored or densely colored section, that's where all the information is integrated. Then in the somas, in the cell bodies of the Purkinje cells, a decision is made. Do we send a signal out to other parts of the brain? And if a signal is sent out, it's sent out via these filaments here that you can see at the top. And we call these axons. And these transmit the signal to other areas of the brain. But now you see something funny here, right? Because when we compare what we see here, we see that there's a lot of branches going on. There's a lot of other little branches of axons going this way. And when we look at the schematic, this is actually not there. So in the last 10 years, a little bit more than 10 years, I've been working on the circuitry of the cerebellum. And what we've actually found out, together with a lot of other researchers, of course, is that this circuitry is really more complicated than we ever thought. And this is based on my own work, how we can refine the circuitry. And you can see that we have found different organizational principles. The cerebellum has specializations over its area. But we can also see that there are more cell types in there. We knew about these cell types, but we didn't know how they're connected. Now, we solved a little piece of the puzzle by filling out a few of these connections. And we even found interactions between neurons that we haven't even dreamt of that they would be possible to exist. Now, this is really firmly fundamental research because it concerns the basic circuitry of the brain. So it's sort of laying a blueprint of how the brain works. But now I'm working at the Wilhelmina's Children's Hospital, where we have more of a translational approach. So how can I take what I learned here in the cerebellum and the techniques that I learned here for circuit dissection, how can I take that and do translational research? At the Wilhelminus Children's Hospital, we're interested, amongst other things, great many other things, we're interested in cerebral palsy. And cerebral palsy is caused by damage to the brain, to the forebrain at an early age. And here you can see an MRI scan of a patient with cerebral palsy. When we zoom in on this MRI scan, you can see that on one side of the brain, there's a lot of damage. So on the right side here, you can see there's a lot of connections, and you can see it here on the rotating brain as well. So a lot of connections between the front and the back of the brain, and there's descending tracts. And on the left of the brain, where the brain is damaged, a lot of these tracts are disappearing. So you can see that here as well. Now, interestingly, this is also causing damage to parts of the brain that are not initially damaged. So here you can see that on the right-hand side in the cerebellum, there's what we call secondary damage. Now, what do we see in these patients? And you can see a movie here on the left of patients with cerebral palsy. You can see that they have troubles moving. They have trouble walking, coordinating their movements. They also have trouble often with eye-hand coordination, and sometimes even trouble with cognition or emotional regulation. Now, what we want to understand is how we can explain going from this damage to what actually we see in the clinic with these patients. So we want to understand what is exactly damaged. What does the damage change to other parts of the brain? So how is the whole brain changed? And what is now the exact relation between what is damaged in a certain area? How is that related to a behavioral outcome? Now, we cannot fully understand this in patients because we cannot do interventional research. We cannot change parts of the brain. We cannot fix this. We can also not just damage an extra part of the brain in these patients. That would not be ethical. We can also not simulate this in a computer because it's too complex and we don't understand even how this system is regulated normally. We can also not do this in vitro or in cell culture tests because then we don't have the complexity either. So for these experiments, we need to use animals to really understand what the relation is between damage in certain part of the brain and a behavioral outcome. Now, this brings a great responsibility to us because we need to take care of our animals. And every day we check on our animals. We see whether they have enough food, enough water, whether their cages are still clean. Actually, you can really carefully look at a mouse and see whether it's in pain. There's what we call the mouse grimace scale. And the animals are checked every day for these signs of discomfort. And especially around the time of surgery, and you can see here a rat that has an implant by which we can record the activity of neurons in the brain while the animal is performing a task or doing some behavior. Around these times of surgery, we of course give a lot of painkillers. All surgeries are done under anesthesia, and we often reward our animals with a little bit of a reward. And here a rat is getting a little bit of a sugary solution reward. So in conclusion...
Sorry, Lawrence, just before you go, I think we lost your screen. Are you able to share it again?
Oh, no. That's bad news. Oh, no.
It sounds very interesting, the slide you're talking about. So,
where did you lose me? Is it back on?
Yes, perfect. Great. Carry on.
Okay.
So just before you started explaining the animal experiment, I think we lost you.
Where is that? Here?
Yes, after this slide.
After this one, okay. So what I just wanted to say here is that we check on our animals regularly and that we take good care of our animals around the time of surgery, especially. You can see here a rat on the right-hand side that has an implant, and these need to be implanted surgically. And around that time, we take good care of our animals, a little bit extra care, that they receive adequate pain killing. We do the surgery under anesthesia, and you can see the rat is actually drinking a little bit of a sugary solution here, which they really like. Okay, let me move to the conclusions then. So in scientific research, we unfortunately still have to use animals sometimes. But this is tightly regulated, and you have to apply for permits, you have to have a license yourself, and the law actually says that you cannot do animal experiments unless you have these 3 criteria. So the first criteria was a scientific benefit that you need to show. The second one is that you need to weigh this scientific benefit and make an ethical deliberation between the scientific benefit and the harm you're doing to the animals. And the third one is probably the most important one. If you can get the same results without using animals, then you are not allowed to use animals. So animal experiments are only allowed when no other means are possible. And then this applies basically to the whole chain. So we start with fundamental research, researching the fundamentals of biology. And then we use translational research to bridge the gap to our fundamental understanding and the clinic. And in both stages, we use animal research. And there's a great responsibility for the researchers that do use animals in research in all of these aspects. And we have to make sure that what we do is ethically sound. So with that, I would like to close. If there are questions, I'm happy to answer them. And I would say cheers.
Yes, cheers. Thank you very much. That was very interesting. So yes, audience, please do not hesitate to ask any questions. I do have a question to start. So you talked a lot about, other models that people would maybe think of, and especially AI. And you said you've been working in this part of the industry for a while in this part of the field. So have you seen advances already that have meant that you can reduce the amount of animals that you would typically use in an experiment? And do you see that actually going further?
I think there are a great many new techniques that are being developed all the time. I can think of one thing that we're using now at the moment as well, and you touched upon AI, and I think that's one of those opportunities. When you look at behavioral research, for example, this has normally been done by scoring behavior of animals. And these things are now more and more and more automated. And this enables us also to do more fine-grained investigations into the behavior. So we're not saying anymore a mouse is walking around or a mouse is grooming, but we can actually split that up very, very carefully in very small parts of behavior. And that greatly increases your explanatory power in any way. And that greatly helps. And actually in the clinic, we're now implementing also an AI technique to analyze movies of patients. And we're using the exact same technique that we use to observe the patients to observe animals that are suffering from the same defects in their brain or the same damage in their brain. So we can also now start directly comparing patients and animals, which greatly improves our applicability. So I think these are great advances.
Yeah, great. And yeah, panelists, if you also had questions for each other, please do go ahead.
I have a question, Lawrence.
Yeah, Daniela, go for it.
Thank you for this nice presentation. I think that with your colorful slices of brain, you really showed how complex is the matter. And also your question is very complex, right? It's a big question. Do you see a chance to cut down this big question in smaller questions, and for some part of these questions to use animal-free technology?
Yeah, this is something, of course, that we're thinking of continuously at the department, and these are really the broad strokes that we want to go, or at least I want to go in research. But at the department, there are a great many different techniques, and we're at the moment setting up a system of culturing neurons in a dish, and then looking what certain aspects or certain treatments would do to that. And this is really work together with Cora Nijboer and Caroline de Tije from the lab, and they're really good at setting up these animal-free methods and really pushing that forward. So it's really great to work together with them. Yeah.
Thank you, Lawrence.
And we've had a couple of questions from the audience. For the sake of time, I'm just going to ask one of them for now, because I think it's also quite relevant to probably the next talk, and then Ali will move on to you. So we've had a question from Janneke Hogevorst from YouTube. And she said there are big differences between human and rodent brains, which of course is one thing that everybody mentions when we're talking about animal experiments. So how do you know that what you see in mice is relevant for humans? How do we make that justification of the translation?
Yeah, I think that's an excellent question. There are big differences. I mean, the human brain is big. A mouse brain is about this big. That's one start, of course. The interesting thing, of course, is that the different parts of our brains we can find back in a rodent brain. So that is really a big similarity between our brains. And we can see the same things back in cats. We can see the same things back in elephants. We are all mammals in that sense. So that really helps us make a good translation. I think, of course, in terms of the intelligence that we have and the intelligence that a rodent have, we have a lot of difference there. So that is something to really take care of. But the cerebellum is really well conserved, especially the cerebellum. Also, the different layers, for example, in neocortex, which we think, which is the forebrain, basically what we still think is what's making you think, that's all pretty much conserved between species. So yes, there are big differences, but there are also big similarities. Now, to figure this also out, what we're also doing, and that has been work that I've been involved in at the VU, is have tissue that is coming from a surgery. For example, when a tumor is removed, they also need to cut out a little bit of extra tissue around the tumor. And then you can really start comparing what you see in the mouse in the tissue and what you see in the human in the resection tissue and see whether they match up together. And what you can see is that there's big similarities, there are differences, but the basic circuitry is really the same. So as long as you keep to these very big strokes, which we don't understand yet, and we have a lot of information that we're still missing, I think you can do great work in animal research without the need for big surgeries in humans, which is very, very rare to obtain that tissue. So it's really helping science along by using the animals as a way of investigating brains, human brains.
Great, thank you. It's very interesting. So for the sake of time, we are going to move on. So please do continue asking your questions. And if we get some time at the end, we will go back to some of them. So I think we can introduce our next guest. So we have Ali Irvine at Nepali.
Ali will talk to us about the use of in vitro models instead of animals and how he can help innovators to overcome some difficulties. Ali, the floor is yours.
Thanks, Natalie. Good evening, everyone. I'm just going to quickly introduce myself. My name is Alasdair. I'm a PhD candidate at Utrecht University, and tonight I'm really excited to talk to you about some of the research that I've been doing and how reducing our reliance on animal modeling can actually help make medical research more effective and efficient. So I work within the Utrecht Institute of Pharmaceutical Science, so I'm going to be referring mainly to the impact this can have on the development of new drugs and medicines. So in order to do that properly, I'm going to quickly brief you on sort of the current practice in drug development and then tell you what my research is about, which is not as exciting as the previous talk, I'm afraid. I kind of do a bit more meta work, it's research on research. And I'll then explain in this context why pursuing animal alternatives is a good idea, and then explain the technology that can go on to replace them. So how are drugs developed? First thing that we have to say, and this is a major caveat, is medical research and development is and always will be difficult, risky, and incredibly expensive. And this is primarily due to the need to ensure safety for patients. When you're trying to create medicines and improve people's lives, the last thing we want to do is create damage or any kind of injury. The second most important thing to understand is this is a partnership. It's a long-term partnership between multiple parties with slightly different outcomes. So as Lawrence has said, drugs, there's a lot of difference between sort of translational work and preclinical work. But essentially what happens is in the drug discovery phase, we throw loads of money, loads of infrastructure, and loads of technology at finding the best molecules, the best interactions, best targets that we can possibly find. And this is pretty much a sunk cost. This is always going to be an expensive process. And then we also have sunk costs in the recruitment of patients, logistics to run multi-center trials, and also for safety monitoring. But at this preclinical phase, before we're in humans, the gold standard right now is animal studies. This is how we make sure that all of the interactions that we've trialed in a cellular model is actually going to make any impact at all in an organism. So animal models really push us through this preclinical phase into clinical development before we start looking at phase 1, which is dose tolerance and toxicity outcomes, phase 2, how well something works, and then phase 3, how well a new compound will work against the standard of care or a placebo. So why should we look at animal alternatives? In recent years, no doubt you've seen health insurance go up. You keep hearing more and more about how much pharmaceutical companies are spending on research and developments, and these drug development costs have really soared, and it routinely costs more than $2 billion to take a drug to market. In 2019, 186 billion was spent on R&D, and routinely R&D budgets will increase by 5 billion a year. By 2026, that can mean almost $230 billion per annum. And for all of that, 80 to 90% of investigational drugs fail in clinical testing. 60% of those failures due to lack of efficacy. So that's the phase 2 trials. That's a very long way through the development cycle before you actually fail. And another 30% due to toxicity. Now, there's quite a few schools of thought about why this is. The first one is low-hanging fruit. Medical science has achieved great things. We've done everything at the very, very basic level. We've basically done everything easy. And now we have to spend more money in order to get to that sweet fruit at the top and actually achieve something quite different. The other thing to talk about is on the far left of the previous slide, that basic research is we have a reproducibility crisis in academic research. We see researchers unable to generate or repeat some of the results to publish papers. But for me, one of the most persuasive arguments is that this sort of translational gap or valley of death is primarily due to the failure of the apex of preclinical research being animal modeling to translate directly into human physiology. You can have drug candidates terminated for lack of efficacy in animals, but maybe it will work in certain subpopulations of humans. Likewise, you can have drug candidates progress all the way through preclinical testing with no issues whatsoever, and then once they reach phase 4 pharmacovigilance, you find out that you have to withdraw everything from the market at huge cost and reputational damage. But what's happened now is, if you remember back to the last slide, the funnel is now as wide as it possibly can be at our drug discovery end and our mechanistic end. So the issue must be somewhere else. This is why, for me, a transition slightly away from a focus on animal modeling as the apex of preclinical testing is vital in order for us to, realize some benefits for patients in the long term. So the long and short answer of why we should look at animal alternatives is the current system's reliance on animal modeling to make the leap into humans isn't working that well. So we need to do something different. Essentially, if we can find a way to pursue a more human-focused research, then we'll get better human-focused response. Ali, I can't believe you're doing a PhD in this. Come on, tell me something that I don't know. For me, this is how I structure my thinking. If we want to build more effective treatments, we have to have a solid foundation. When we think about alternatives to animal testing, we're always talking about refining the use of modeling, reducing the number of models that we use, or indeed replacing them. And then for me, we have to evaluate these alternatives on an ethical, economic, and scientific basis. So how do we go about finding better models? Part of my research is doing exactly that. And this slide shows a sort of a summary of all of the different models we use in the preclinical stage to test the safety and effectiveness of new medicine. These models of sort of biological tissues are indispensable tools for unraveling both human physiology and how diseases work. And they usually consist of a single layer on a hard artificial surface. So if you think, when we say in vitro, Latin of the glass, so essentially one layer on the bottom of a receptacle. So this makes them pretty robust and suitable for parallelized high throughput research. You can test hundreds of compounds in one go in hundreds of different cells. But due to their simplicity, high throughput, it's not particularly physiologically relevant. It doesn't look like the human body, it doesn't act like the human body. And so even though the realism of these models can be increased by including more cell types, it inevitably leads to this kind of decrease in robustness and throughput. And this is a constant trade-off that we see between realism and simplicity, and this leads to a bit of an impasse in the development of new in vitro models and new techniques. But you might notice something. I'm highlighting this type of model, an organ on a chip. And this is a really exciting new class of preclinical model on development right now here in Utrecht. So what are they? Why have I put up a yin and yang symbol? Because organs on a chip as a class of micro-engineered in vitro modeling have the potential to break that in vitro impasse that I talked about earlier. This is a pretty exciting technology, and every time that you hear someone talk about it, they'll say it's got the potential to revolutionize disease modeling, drug discovery, and toxicology by strengthening both the relevance of culture-based modeling to a human body and also reducing our reliance on a costly animal study. It's essentially a combination of an artificially engineered, physiologically realistic microenvironment. So it might not look like a human when you're looking at it, but on the micro scale, this is what it absolutely looks like. But because of a combination of new technologies, we're able to both allow this high throughput and indeed keep what is essentially a human microenvironment, so an interface that could be blood vessel to kidney, blood vessel to lung, exactly the same. But I said chips. So not chips as in Scottish chips or chips as in American chips. I need you to think in terms of computer chips. And these systems can replicate the kind of flow and diffusion of nutrients, waste products, and drug metabolites to give us a more detailed and realistic model of a human response to a therapeutic dose. And we can incorporate a whole number of tissue sources with the potential to include patient-derived materials from sources such as the urine and feces, and take these stem cells to benefit and make a more robust, long-term system. And the ideal end goal is a microfluidic body on a chip, allowing for the study of complex systems interaction and disease through a hyper-controlled system. Now, that's the ideal scenario. How is this possible? It's only really made possible by the convergence of multiple technologies and research disciplines. It's a hard thing to do, but this sort of confluence and convergence takes ideas from stem cell biology, including the idea of induced pluripotent stem cells, mixing huge advances in cell culture capabilities, genome editing, 3D printing, engineering both on the micro and macro scale to create sophisticated cell sensors within the system. And what we found is, and demonstrated, is that a dynamic culture condition, even with a traditional cell line, will increase the physiological maturation and function of this system. Now, I've talked briefly about this bridging the gap, but organ-on-chip platforms also have can use patient-derived stem cells, and this can be used later in the clinical trial process, almost as a personalized medicine approach. So this can be used for in vitro therapeutic testing before actual dosing with a new compound in hospitals. Huge potentials. And in the US now, they have a research institute dedicated to clinical trials on the chip. But what does it look like? Oh, look at that. That doesn't look like a human either, Ali. But the point is, it does on a micro scale. Okay, you will often hear organs on a chip referred to as microphysical systems. It's very early stage technology. I mean, this looks like something that you would make in the back of your dad's garage with your soldering iron after physics club or something. But by pursuing this field, we can hopefully change our brute force R&D methods, which is now described in the forms of something called Room's law, which is one innovation roughly every 18 years. There's something closer to Moore's law of microprocessors. So that's an innovation every 2.5 years. So my research focus, it's not that exciting, but hopefully this brief overview is giving you some insight into the potential of organ-on-chip technologies. And where I fit in is I'm not a laboratory researcher anymore. I used to work in clinical research. I was there for 4 years. But I kind of hung up the pipette now. This is where all the exciting stuff happens, down here on the left-hand side. All of the work with the cell sourcing and all of the high-tech engineering. What I'm talking about is quality, study design, regulations, and how we can actually build solid foundations and strengthen our arguments for 3 Rs. Why is this important? this is supposed to be fun. We're going to talk about regulations and standardization, validation over pints. I know, right? But let me explain. I briefly touched on the factors affecting R&D at the start. And if we keep the bones of our current ecosystem, we may, in fact, actually repeat some of the same mistakes, essentially creating another reproducibility crisis. So when pursuing an alternative to the current system, we've got to build on solid foundation. So if we take that as read as animal alternatives, because the current system isn't working, then we need to also, if we're just throwing new technology and vast amounts of money at the same problem, we need to make sure we're building an appropriate system. And this is something from a paper published last year that really, really concerned me. Those developing and those who are most likely to use this sort of technology do not understand the regulatory environment in which they want to operate. So this is where I come in. And there's so much exciting work coming on in Utrecht. However, I can't just be a homer from my own research institute. I need to kind of be agnostic as to where the stuff comes from. We need to find the best stuff and evaluate it properly. I've been working on a series of systematic reviews, and we're looking at a focus on the major organ systems involved in the initial clinical evaluation of new drugs. So we need to understand how drugs enter and are processed by the body. And this field is known as pharmacokinetics, and it aims to quantify processes of absorption, which is why the gut is there, the liver is there, the absorption, distribution, metabolism, and excretion of new compounds. These are the key organs, right? So intestine, liver, kidney, and how compounds move between them. So my work worked on a creation of a framework to evaluate these in a scientific way, an economic way, and an ethical way. And then from there, we can identify high potential models and then we can schedule them into the right R. So whichever it is, reducing animal modeling, refining animal modeling, or replacing animal modeling. So the scientific arguments are pretty clear. More human cells due to increased stimuli and the 3D nature. On a micro scale, these cells look more like humans. It allows us to find control of the microenvironment. We can test and take so much data from these systems and then transfer that into clinical practice. And it allows us this ability to integrate sensors and new technology. Some of the economics behind this. There's huge potential, but I really want to say this. It's like the potential has to become almost $117 million market by 2022, while ensuring a 26% reduction in R&D development costs. Now that's $650 million per drug candidate. And to put that in perspective, your average European healthcare system will reimburse you for 35k per quality adjusted life year. So for every year that a medicine is going to give you of life, the health system will pay about 35,000 euros. And this is difficult. Innovation in this space is not going to be linear. It's not going to be like the drug development process in a straight line. It's very much going to take feedback, multi-parties, and a number of stakeholders involved, especially regulators. And most important of all, patients got to be front and center. I said this has a potential to inform personalized medicine and personalized drug profiles, but the only way we can do that is we keep the patient front and center. So some challenges. My work very much builds on some of the outlines of strong foundations by finding relevant endpoints, reference drugs, measurement methods, and parameters to assess organ functionalities. But there are huge technical challenges, not least linking separate chips to in a physiological relevant way. And I just talked about saving billions of euros, billions of dollars. But the one thing that can really undermine a microphysical system or organ chip is bubbles. And we also have to consider creating some kind of universal media, a blood replacement, in an animal-free way. Lawrence, we also talked about before, fully understanding the mechanisms of action of complex diseases is very, very difficult. And as I said, we can recreate the microenvironment of the human body, but how can we recreate the microenvironment if we don't understand the microenvironment? So there is strong arguments for, but there's also arguments against. And one of the things that I found not disappointing, but quite surprising was there is strong institutional memory in academia, as well as in some of our pharmaceutical partners. The number one reason that someone would choose to use a model, whether it's a 2D culture model, 3D, or whatever it happens to be, is because they've always used it. So tradition, not scientific, not economic, not ethical reasons, is the reason that people will choose what model they use. And then the other thing to talk about is Gartner released a report in 2018. I'm not sure if people understand who Gartner are, but they called organ-on-a-chip technology the hype boy of the 21st century. So we need to understand the limitations of technology. Which are we actually aiming for? And there's a bit of an elephant in the room here. I'm talking about creating these frameworks and these, everyone in a multi-stakeholder environment is showing the different groups. There is value for everyone in applying this approach. Potential end users have already invested heavily in the status quo, and there's lots of old economic truism, but we really need to find out is, will our arguments for implementing animal-free research methodology be persuasive? And do end users have the same perception of a good model as innovators, as people like me working on trying to get these into the market? So these are just my final takeaways for you guys, is the application of animal alternatives can provide a pathway to sort of science our way out of the R&D and reproducibility crisis, but the new ideas need solid foundation. Standards are important, we can't keep making the same mistakes. There's a need for new ways of doing business, and the biggest hurdle is kind of showing everyone that this is a valuable way to do things. There's hurdles in institutional memory, and first, if you remember back to the middle slides there, we're not there yet. This is early technology, but there's some exciting times ahead, and I'm really happy to be working in this field. And that's I'll let go.
Thank you, Ali, so much. That was an amazing talk. You did a very good job pointing out the most important values and the biggest challenges in the field. Before we shift to the audience's questions, I have a question for you. It's a relatively simple question, but do you have an example of an in vitro model or a test that actually substituted a routine animal testing method in either in research or in drug testing?
So we've seen great progress in the cosmetics where animal testing was banned, and we do have some very exciting skin on a chip models. And now the FDA has clear guidance on what you need to show if you were to include organ on a chip, what they call it microphysical system data within a regulatory binder. Now, regulatory binder is about this big for it, but there's very clear pathways that we can now pursue. So yeah, I think the skin and cosmetics is the first one, and then we can kind of follow on as we progress.
Thank you.
Yes, very interesting. Yeah, I was going to say just before we move on to our next speaker, we did have an interesting question from YouTube from Very. So she said, human beings are capable of pain, which this chip is not, as they lack neurons and mental capacity. Is it ethical to use these drugs on humans before testing this on an animal model? And I also just wanted to add, obviously we talked a lot, and as you said, this is very new technology. We talked a lot about the microenvironment, but what about when we're talking about the biodistribution, so things that happen in other parts of the body that maybe you're not quite expecting. So do you see these chips being able to tackle these problems?
I think the best way to answer that is to say, from an ethical point of view, the only way to replace something in a medical system to prove it is just as good or better than the current practice. That is always our validation guideline. So if you believe in open science, like I do, the idea of having the gold standard published animal data, and then we can prove that doing it this way or using this sort of technology is just as good is the only way to prove that. So from an ethical point of view, I can't say yes because I don't know yet. But what I can say is if we follow this pathway, there's a potential that we can also undo the moral dilemma, which is what we're using right now clearly isn't working either. So we have to try something new.
Yeah, definitely. That's very interesting. Great. So we are going to move on again, just for the sake of time, to our next speaker. So we have Dr. Cyrille Krul, and I think that she's going to maybe kind of discuss some of the ways in which we do transition to animal-free testing. So please, the floor is yours.
Yeah, thank you so much. And thank you for the previous speakers, because they told you already a lot of what I also would like to touch on. But I hope I can explain you a bit more what the transition is we are making from let's say, the 3R policy to animal-free innovations. And I think Daniela will continue later on with how we are doing that in Utrecht. Yes, as introduced, I'm working at the University of Applied Sciences in Utrecht. And so that means that our work is really focusing on how to use those animal-free innovations in, for example, drug development, but also chemical safety, etc. Briefly, to give you any idea where we were talking about, and I think it was already said in the introduction, almost 500,000 animals are being used in the Netherlands every year, and this is more or less stable since 2000. So from 1917, when we started at 1.5 million a year, it's now 500,000. So it's a nice way. We decreased the number, but now it's more or less stable. So we have to do something different. And also in the EU, around 10 million animals are being used, mostly mice and rats, as already said. And worldwide, we can only guess, because not every country is calculating or counting the animals. But we expect that it will be between 100 and 150 million animals worldwide. So quite a lot. But you might think, where are we using it for? Laurens showed you the figures. I think I don't know whether it were the Dutch figures or the European figures, but they are quite the same. So indeed, most of the animals are being used for fundamental research. The second largest is for safety testing, and every regulatory experiment that is needed for introduction to the market, etc. So also this changes a bit every year, but is also more or less the same every year. And the nice thing is that in 2014 we started to record and count the number of animal experiments in life sciences in whole Europe in the same way. So actually now we are able to compare also the data. Yeah, we were talking about alternatives to animal testing and animal experiments, but I don't think it's really a good term to talk about alternatives, because the alternatives might give you an idea that it is something less good than an animal experiment, or it also means that you are comparing it with the animal experiment. So the animal test as the gold standard. And I hope I can show you that we really need to look in a different way to this to really make the step to animal-free innovations. So I think Ali just said it already. There are reasons not to use experimental animals. As shown in the first by the first speaker, it's not that easy to do an animal experiment. It's regulated by the law, and it's very good. And but from scientific point of view, we know that there are also limitations in the predictive value of these animal models for humans. And also, as explained, for many research questions, you would really like to know the mechanism. How does it work? And to predict whether it would work in the same way as in humans. But it isn't that easy to do that with rodents. And during my PhD, which I have to say is already a time ago, I realized already that, for example, in the GI tract, the physiological conditions in a rodent are completely different than in humans. And that brought me also to different conclusions and different results. And of course, there are also societal and ethical reasons and economic reasons. The animal experiments are very time and money consuming. And some companies also, from a corporate social responsibility view, don't want to use animals anymore. And we touched upon it already very briefly. For example, the cosmetic directive in Europe, but in Netherlands it was already since the 19th forbidden to do animal experiments for cosmetics. There are quite a lot of regulations looking for other things than animal testing. So the 3 R's, refinement, reduction, replacement, is really not new. Bill Russell and Rex Birch already in 1959 published their paper about the principles of humane experimental techniques. And there they introduced already the terms 3 R's, and the refinement, reduction, and the replacement of animal testing. And they demonstrated already that if the animals have less stress and are less painful, you will obtain better results, more reproducible. So they really stimulate researchers in all the fields to look at these 3 R's. And actually all the legislation in the Netherlands, in Europe and worldwide is based on this 3 R principle. And so refinement means indeed pain reduction, but also age enrichment, with respect to the social housing of animals. That's very important. Also, the number of animals can be reduced by making use of imaging, for example, or statistical designs where you need less animals than per group. We also talked already about replacement by in vitro models or in silico computer models. There's a lot of terminology going on in this field. To give you any idea about what we are talking about, from the right to the left, and apologize for some big words, but so in Cerebral means actually expert judgment. A lot of knowledge and expertise is in the brains of people, and you should really make use of that. Of course, the computer models, the PBPK models, the models where you can simulate the physiological conditions, as Ali was just explaining, that is more and more also be done by computers, but also in vitro cell culture models, 3D tissue models, etc., with primary cells from animals or from preferably from humans. Ex vivo models, where you can take out, for example, here you see a picture of a rat liver, which you can use outside the body for a couple of hours and sometimes even days. Of course, the in vivo model and also in vivo experiments in humans. We know, of course, the clinical experiments, but also, for example, occupational safety assessment has been done in human volunteers and workers around to see what the impact is of exposure to chemicals. And there are many new techniques, and the developments are going very quickly. A couple of these technologies are already mentioned, the stem cell technology and the organs on the chip that are being developed, sometimes based on these stem cells, but also some other technologies that are not invented as an alternative to animals, but that can be used in this field. So, for example, e-health technology, so data that from humans that can be obtained and can be used to have any idea about the physiology of humans. 3D printing of cells, but also of medicines. Of course, we talked already about the data integration is very important. Imaging, but also something like microdosing, which is a technology where you can dose humans at a very low dose, 1,000 or even lower of the therapeutic dose being used in humans. So these technologies are developed in completely different areas by people that are not usually not involved in animal experiments. And I think it's very interesting to bring those people together, people that are working in a different field and being inspired by the possibilities for these technologies, and see how we can use that in answering the scientific questions we have. Yeah, I think that there's a lot of going on. Here, for example, a model which is a gastrointestinal model simulating the processes in the GI tract, which is made from glass and flexible walls inside. And in this model, you can really simulate the digestion, which I explained to you already, which is completely different in rats and mice than in human. And especially the pH, for example, that can be simulated in a way how it is in humans. And you can also simulate the situation, for example, in babies and very small children, and in elderly, where also the process in the GI tract is different. And I worked with this model many years ago, and it's still being improved, and cells are being introduced in the model, and also the human microbiome, for example, so that it looks more like the human situation. Organoids, we talked about it already before. So these 3D small organs are being cultured, which is quite difficult, and difficult than the cell culture we did in where we start with in plastics, etc. And in these 3D structures, you can also simulate the physiology in humans in a better way. And there are thousands of models that are being developed. But of course, it's going not that quickly. The use of those models, as explained by Ali, the validation, the acceptance, the predictivity of such a model. There's a lot of research needed before we can use that in a broad way and for many different domains. So we, as said, the Dutch government is really promoting this transition to animal-free innovations. And actually, we want to do that in a way that we can improve the scientific research, but also the product development and personalized health, which was also mentioned before, for better treatment for humans. And Daniela will explain you in the next presentation what's going on in this program in the Netherlands and also in Utrecht. Personally, I believe that if we want to make that step to more human-based models, models based on human cells and human biology, we could also facilitate the researchers. For example, the use of human cells, making use of human cells and human tissue. Every researcher would love to use that, but it is quite hard and difficult to get that material. For researchers that are working closely together with in an academic hospital, for example, they have access sometimes to this fresh human tissue, but not all researchers. So we have stored material, but that's not metabolic active anymore. So if we really want to make use of human cells and human tissue, we should try to set up a system where these type of materials can be brought from the hospital directly to the researcher. And Vital Tissue is an initiative which is started with a consortium from academic hospitals, organizations like the Bloedbank, the Blood Bank, Sanquin, and researchers also from the pharmaceutical industry that are trying to set up such a system where the leftover tissue from surgery, from patients, can be donated for research. And I think that would be of a great advantage for researchers all over to get access to this material. So it's something different than donating an organ. That's not. It's really leftover tissue, not used for diagnosis anymore, and which is now thrown away. And that is very helpful material for researchers. So I hope we will be able to set up this in the Netherlands in the coming years. We started already with a feasibility study, and that looks very promising. And that's bringing me also to the last example. I would like to show you that from the 1st of June, we will start with a large project where we try to combine all the things which has been said before. So we would like to build a virtual human platform. In this case, we are going to use that for safety assessment of compounds. And there we will put together all the data that are available. So human physiology, biology data, data from experimental models, from those nice advanced human models, and put that in a platform where these data are connected and where AI and very advanced data science is being used to predict what happens in human. And we there focus on situations that cannot be simulated or very difficultly simulated in animals, to get more data on the effects of compounds on humans. And this is a platform which we are going to build not only in Utrecht and also not only in the Netherlands, but with also partners in Europe. And such a platform that should be growing and feed data in so that we can improve it in the coming years. So we started for 5 years, and we got a large grant for this, and I hope we in the coming years we can set up this platform. So that brings me to my last slide. So a question that is often asked to me are we going to move away from animal experiments in the near future? And actually, I have to say, I don't know what the near future would be. How long does it take before animal experiments might be redundant? If we have such a platform as the virtual human, that might be possible. But what I know is if we want this and try to move further and faster, we should collaborate, we should coordinate all the work, we should have commitment from all the stakeholders. So not only the innovators, the developers, but also the end users, the regulatory authorities, etc. We should communicate about what we are doing. And I published this a couple of years ago as from the 3 R's to the 4 C's, and there are many different, many more C's already explained. The people were saying to me, okay, this is a very nice idea, but that means that we need confidence in new models, new way of working, and also that we have the competence, so that we educate people in this way, that such animal-free innovations are feasible and accessible for everyone. So I thank you for listening, and I hope you are inspired to work with all of us here on this animal-free innovations.
Great, thank you very much, Cyrille. That's very interesting. And yeah, I just had a quick, maybe it's more of a comment than a question, but the vital tissue is, I assume, maybe works at the moment on a opt-in basis. And so should it be like the organ donation where it's on an opt-out basis, where the norm is for it to be going to research instead of being thrown away? and people have to opt out for it to be thrown away instead. Is this maybe a way to push forward a program such as this, do you think?
Yeah, I really would love to. Yeah, that would be my dream, of our dream, that it is in the same way as the organ donation. But now it's still, we need the informed consent of the patients, and that's fine because the people are really willing to help. But for the future, it would be good that everyone can somewhere address that if I need a surgery at any time in my life, I'm willing to donate my tissue. That would be great.
Yeah, should be just the next box on the form, right, of the organ donation. Why not?
Yes.
So we will quickly move on, just so that we don't go too far over time, and address any questions at the end. So Daniela, you're going to be our last but definitely not the least speaker to, I guess, continue this conversation that Cyrille started to discuss the transition towards animal-free testing. Please go ahead.
Thank you, Anna. And I can move the slide from my screen. Let me see. No, I think just a second.
Yeah,
yes,
we can see your slides.
yes,
So no problem.
yes,
yeah.
I understand. Good evening, everybody. And I was really inspired by the talks before mine. And my topic is very much linked to what my colleagues have just said. We think about a world without laboratory animals, but how to make it happen. We have discussed about gold standard. We have discussed about institutional memory, about working with animals. How can we change this mental framework? So during my presentation, I'm going to talk about the transitieproof die vrij innovatie in Dutch, but the transition to animal-free innovation from the government and what we are trying to do at Utrecht University. Who is TPI? TPI, Cyrille already introduced it, is coordinated by our Ministry of Agriculture, Nature, Food Quality. And the TPI is a sort of partnership with many organizations. So Cyrille talked about the 4C. So you see that also TPI is really based on communication, collaboration, and competencies. And you see here in this slide that there are a lot of partners, from Association of Health Funds to other sector and also funding bodies. The partners behind TPI are also society, academia, and the business community. In order to make the transition happen, we need not only good ideas, but we need money and collaboration. This topic, if you have followed also during the COVID situation, yes or no use of laboratory animals, arises a lot of emotions. And I do think that we are quite far in the transition to allow us to have a very nuanced discussion, an inclusive discussion, where also the differences are important. And basically, the TPI strives for the best possible outcome for both humans and animals. Of course, nobody wants to deny that the animals have helped us immensely, that from the experiments in the past, from animal studies, we have gained a lot of knowledge. But there are also a lot of bottlenecks. You have heard from the previous presentation. So yes, we would like to have more patients that are treated with new drugs in a very short time. Now, inspired by the government and by this mission to have the best possible science significant for humans and animals, but with non-animal experiments, the academic community at Utrecht University decided to create TPI Utrecht. Now, I am the chairperson, but this is really a very cohesive group coming from Utrecht University, from the Academic Hospital in Utrecht, and from the University of Applied Sciences. And together, we have some core values. We believe in one welfare and one health. We strive for top research and education quality, for sustainability and mutual trust. I would like to cite a sentence that Hank Smith, the chairperson of the NCAD, said some time ago: "We do not blame people, but we like to add something new." In other words, we want to work together. We want to share our knowledge and work together towards new knowledge, and being able in a constructive way to go through this difficult and engaging topic. I would like to bring you through the concept of target images approach. I took this picture from the website of the Netherlands National Committee for Protection of Animals Used for Scientific Aims. So how are we gonna start this discussion? For many research scientific topics, we are still using animals. How can we pose the question, and starting from this question, investigating if there are possibility to go for animal-free research method? How can we do this? We strongly believe that the only way to do is to join forces within our research domain, but also with help of many partners. And we already mentioned the private sector, the business sector, and all NGOs. We need to start this discussion together, because this is really a way to reflect how can we reach the animal-free goal, with possibility do we have to work with less or with no animal experiments. Leaving the idea of animals as a gold standard, it takes a lot of effort. So only an integrated approach can bring through this way. And yeah, it is a step-by-step plan where we need to be able to define the challenges. We have also to be able to investigate the possible innovation. And yeah, we need to have been able to make a risk-benefit analysis, and at the end to come up with an innovative strategy. Now, a lot of words, right? You know, a lot of good words, a lot of positive words, but how can we make this very practical? At Utrecht University, we have created also a website where we can convey also this initiative. For example, we have a contact TPI help desk where the researcher can pose their question, like we are doing tonight in this chat. This can be constantly done, and our TPI group can direct the question to the expert within and outside our university. We strongly believe that the animal part and the animal-free part, they need to work together. And for example, the Animal Welfare Body is an important institution that works on the 3 R's that Cyrille has just explained, that it could be a fantastic motor block to connect animal and non-animal research. we have constantly news on our website, reporting, for example, the latest findings in animal-free research, but also the most interesting conferences going on. We have also worked in the last year, and the new helpathons will come during the next months. Here I show the picture of a colleague from the UMCU. This is a professor that received last year a VC grant for studying liver diseases, and in this in her research domain still animals are used. So what is an helpathon? We closed ourselves for more than 24 hours in a room asking questions. How could we reach the same research goals with less or with no animal? So now this reflection time is something that, yes, we call helpathons, but that we would like to have constantly within our community. And yes, we talked a lot about research, but what about education? If you look on the right side of my slide, you see the first page, the cover page of the last report of the European Commission about how to improve education around the 3 Rs. The outcome of this report is that we need to be able to introduce the 3 Rs as early as possible in our educational programs. And also here we are trying to work hard. We had an helpathon last year in order to understand how we could give the best education about the 3 Rs. And this year we had in Utrecht the first TPI elective master course. We would like to expand this idea of a new education program much further, and again much earlier than the master program. For example, we would like to start at the bachelor level, continue with the master level, and then with post-academic training, and linking all students from all faculties, because we think that this is a transdisciplinary approach that really is needed to make the change. So we are working very much in the last period to create a framework where education about the 3 Rs will be given very early on in the curricula, but also in a consistent way throughout the curricula. Perhaps you know, Lawrence talked about animal experiment. There is education dedicated to how to perform experiment with animals. We would like to start very early on with education about the 3 Rs and the replacement of animal experiment, and more also connecting different type of new and advanced technology. Yeah, we talk always about research, and you saw that for this country, the percentage of animals that are killed for education are lower compared to animals that are killed for testing or fundamental research. But do not underestimate that in Europe, every year, around 1 million animals are killed for teaching clinical procedure. So for training. I am myself a veterinarian, so you see on the left a picture that is taken in our dissection room. At the moment, we still rely on very classical technology for teaching anatomy and clinical procedure. But now with virtual reality, with all digitalization of our education, also there we can make the difference. And again, we should try to make this difference together, connecting our possibilities. So you see that now we work in large network, in multiple systems. So I think that the challenge for the future is how to connect in the best possible way these different systems. And TPI Utrecht would like to create this possibility to be connected in a very constructive way. And I think that this is my last slide, and I would like to thank you very much.
Thank you so much, Daniela. We are very excited to see the initiative. Also, I was very impressed to see the educational level. Actually, I have a question for you. That was actually touched upon by Ali. Do you come across researchers or scientists that are a bit hesitant to try new innovations based on in vitro models, for example, because they are so used to having animal models, because they're used, they are traditionally used, and they are not so open to new technologies. And how do you create incentives for these scientists?
Now, I don't think that I do think that there is a strong debate about this topic, but I also do think that we are quite far in the transition. Lawrence, at the beginning, he said, yes, I have a fantastic group of colleagues that is working on in vitro models. We are not there yet, but we are working on that. And as a veterinarian, I read a lot of animal experiments, project proposal. And you see when you read the project proposal that even when they use animals, there is a large part of the work done in vitro and really focusing on new technology. Now, your second part of the question, how to make it happen. We really need to work in consortia. We really need to have this very large partnership. Cyrille has showed a recent consortium, the Human Health Platform. She has showed the human vital tissue. You know, this is a concertation of activities that they come together, and they need to be also as a management embedded in what we do. And I think that the challenge is really a governance challenge. How can we make it happen? It is not only about science. It is also how to connect to what we do, how to connect this very good idea to what we do. And that, I think, that it will be the challenge for the future.
No, I totally agree. And this was maybe touched on a bit earlier by one of the comments from the audience talking about, what would happen in the next pandemic? Are we ready regulation-wise? But I would maybe even like to expand on that a little bit to talk about, if we look at this pandemic that we've been in, and for example, the mRNA technology, how this was something that was maybe not widely used, but a little bit there. Do you think it could possibly take something big like this to drive forward some of these alternatives that we talk about, like organ on chip, to make people collaborate more to bring this further at a lot faster pace? And I guess this is open to any of you, maybe.
I can give my opinion first.
Yeah, go for it.
For me, it's not necessarily what people would like to do. It's what people have the time to do, the resources to do. So if we want people to implement this kind of principles, then we need to sort of incentivize. I remember I tuned into, it wasn't quite a TED talk, but it was one of these scientific mindfulness things. And there's this graphic of people being pulled in two different directions. And it's like pressure to publish, pressure to back up and report all your results properly and stuff like that. So in terms of the next pandemic, pushing people to do it, I think people would want to do things differently. but I'm not necessarily sure that people have the time to do things differently, which is so I'm not sure if it's a willingness thing, or so I'm not sure applying more pressure would make the change. Perhaps letting pressure off and allowing people to do the things that they want to do might make a change. But that's just my opinion. I'm not sure if there's any stats to back that up, I'm afraid.
I personally don't believe in putting pressure. I think that the change has to be owned, right? Also, why do we use also this way of communicating, this way of working around this project? We need to be the owner of the question. We need to believe that there is the possibility to change and that the change is important. I personally do think that the change is deeper than perhaps we can appreciate. If we talk with our students, that they are much younger than I am, or of my colleagues, you see that they are already so critical. And when I was working with the master elective TPI, they had so many questions. They worked out so many innovative way of thinking around the problems that now we face with the animal experiments, yes or no. That I feel positive that if we will invest strongly also in education, we can create a new generation of researchers that from moment one they will embrace problems in a different way.
Can I say something about that? Because I feel that is not really the complete story, I would say. Because when I talk to researchers also that work with animals, I never see them say like it's the gold standard, so that's why I'm doing this. It is always the question that is leading. It is always the scientific question, and then you're going to find which model fits best. And if that's an in vitro model, for example, we've seen some really nice examples of organs on a chip and the gut that Cyrille showed. I think if you can solve your questions with those models, it's not even wise to do it. You're required to do it. There are other questions that you cannot solve with those models. And I think then the fundamental question is, do we want to solve these questions now? Do we want to work on them now in animals? Or are we going to say to patients, no, we're not going to solve this because we don't want to use animals for these questions? And that is the ethical deliberation that is often missing. It is often putting the animal research versus the scientific progress. And it's never saying like, is it ethical to postpone figuring something out and harming a lot of patients? That ethical deliberation I have hardly heard. And I think that's a shame.
Now, Lawrence, I do think that you are right. We need to choose the best method that is able to answer in the best possible way the scientific question. But I do think very strongly that there is the possibility to believe that in the future we can have much more advanced organs on chips that they could be used to answer certain questions. I think I do think that this discussion, it would be a pity if this discussion would tend to put the researchers that are using animals in a corner and to blame them. That is not, I think, absolutely the aim, and it is not even how our government, I am not Dutch, but our Dutch government puts it, because the Dutch government has not banned animal research and has not said that we need to stop with animal research by 2025. So I do think that we should really totally avoid to create this polarization and to work together.
I think so.
And to add to that discussion, I think it's very difficult to say animal use in general. So it's really depending on your question and the models available. And you can't say, if I cannot do an animal experiment, it will stop your research on that disease. That's also not fair. Yeah, so I agree that we should not polarize it and keep really the nuance in this discussion, which is hard. And we also have to explain, because you were saying we are not taking the animal experiment as the gold standard, but there are reviewers of journals that ask you to repeat a result from an in vitro experiment in an animal experiment.
Yep.
And as long as we keep that and say, okay, then I will do my animal experiments, then we keep that going on. So it's also that the researchers, and that's of course very difficult if you are at the start of your career and then say, no, I'm not going to do that because I trust my in vitro data and I'm not going to repeat it in an animal experiment. And it means it might end up that you will not have that very nice high index paper. And that's a very difficult discussion, but that's also something we have to discuss.
No, these models need to be validated really carefully, I think. And I think there's...
but how should we validate it if you don't want to use the animal because you develop something different because the animal model was not giving you the right answer? So then you cannot validate it with the animal model.
Well, it's always a model, right? So
the human itself, hopefully.
So,
well,
Ali,
that's
you just wanted to put a comment?
what you're trying to get to.
I was just going to say, because there's also, I think Cyrille put it really well. It's like there's the publication pressure, and then there's Boris just to say, I don't think we'll ever quite be able to completely model the human brain. That's just, yeah. I think there's a really good point in the YouTube comments about sort of increasing transparency and making sure that data is available. So it might not necessarily be able to comparing an in vitro system directly to an animal, but it might be that there is animal data and there is databases and like data on data that we can compare to as the gold standard. Because if you think about transitioning into a clinical trials environment, then the apex of our medical evidence pyramid is always a meta-analysis of a systematic review. So therefore, we should also be able to do the same for sort of surgical interventions in animal models and really be able to do it that way. So it's not necessarily about direct comparisons, but it is about data comparisons. And then it comes back to these strong foundations that I was talking about transparency, reliability, and producibility, which also, for me, flies into the idea of appropriate model selection.
No, definitely.
Yeah.
I think it's very valid. So, sorry, Lawrence, we'll do one more comment, but we will have to wrap up because we're going to
let Lawrence go. He's been holding
Lawrence, go for it. Yeah, go for it.
Okay. So I think that this validation of models is really important. I also think that the example that Cyrille mentioned, that the rodent gut is really different from the human gut, that tells you something about the applicability of that model for the human gut. I try to show you that the rodent brain is actually quite similar to the human brain, so I think it's a good model. I think we also need to realize that the in vitro systems and the non-animal technologies are models, and they have their caveats, as do animal research. And I think that's also why this openness is so good. And we're also trying to do this in animal research. And I think the University of Utrecht has some really great initiatives in this direction for preclinical research. In fundamental research, this is much more difficult. But I think, this openness, and I think we see this across science, from psychology to medical science, and that can really help us forward. And I don't think this is something special to TPI or to the non-animal techniques that we've been talking about tonight.
Definitely. So yeah, we're going to have to wrap up now because obviously we're a bit over time, but I just want to have a huge thank you to all of you and the speakers and to everyone who participated. Some really interesting comments going on in the chat. And yeah, it's a conversation that needs to continue happening, and I think will continue happening, and we'll be seeing a lot of technology advances and pressures from all different spaces. And yeah, I think it's very, very important that we keep this as an open conversation, as Lauren said. So thank you again to the audience for listening in. And yeah, we hope that you will carry on enjoying the Pint 21 events. So I think tomorrow night we have an event on physically distant, socially close, talking about the intimacies of social touch technologies. So please do tune in tomorrow night to see the last couple of events and on Friday. And yeah, so again, thank you to our speakers and good night, everyone. Cheers.
Thank you.
Thank you.
Bye.